Hello, everyone. I'm Patricia Kaufman, the co-founder and communications director of the CLL Society. Welcome to our 6th COVID-19 virtual community meeting, the Delta variant and staying protected. As we enter the fall of 2021, the COVID-19 pandemic continues in the United States due to the increased prevalence of the Delta variant, coupled with suboptimal vaccination rates. It is critical that CLL patients and their caregivers continue to receive up-to-date, credible information from expert voices. In our continuing COVID-19 virtual community meeting series, we remain dedicated to providing helpful information to empower our CLL patients and their caregivers to make safe and informed decisions. Smart patients get smart care. Later in this webinar, our audience can engage in a question and answer town hall format with our expert panel comprised of 2 CLL experts, an infectious disease specialist, and a virologist to discuss the current COVID-19 challenges affecting our CLL patients. We would like to thank Adaptive Biotechnologies, Bristol Myers Squibb, Janssen, and Pharmacyclics for supporting CLL Society's COVID-19 virtual community meeting. At this time, it's my pleasure to introduce our moderator, Dr. Brian Kaufman, Executive Vice President and Chief Medical Officer of the CLL Society.
Thank you, Patty, for that nice introduction. I'm Dr. Brian Kaufman, Executive Vice President, Chief Medical Officer, and with Patty, the co-founder of the CLL Society. And I want to welcome our audience and the incredible panel that we have today for today's event. But before we get started, I want to, with the panel, I want to have you spend a few minutes with Robin Brumble. Robin's a nurse with a background in education who serves to direct our scientific affairs for the CLL Society. And she's put together this incredibly helpful tool, kind of an action plan, some guidelines for all issues related to COVID, kind of preparedness packs. So Robin, it's your floor now. Thank you.
Thank you, Dr. Kaufman. Hello, everyone. I'm going to review CLL Society's COVID-19 action plan with you today. Many of you in our patient community were reaching out to CLL Society after either known exposure to COVID-19 or receiving a positive test result. Some even voiced that they weren't sure what to do and asked if there was anything that they could have done differently or done something to be more prepared. As a result, CLL Society decided to put together a COVID-19 action plan, which is comprised of several checklists that you can begin to fill out right now before COVID-19 impacts your home. We highly recommend that you spend some time reading through the directions, filling in the blanks as they apply to your specific situation. Print out these completed checklists and keep your personalized plan in an easily accessible place where you can refer back to it should you ever need it. I'm going to go through the 5 pieces of the plan and provide some screenshots very briefly. The first 2 pieces include reading through the step-by-step directions for completing the plan, and then filling out the corresponding checklist called Complete Prior to Exposure. The intent of these 2 documents is to guide you through what exactly you need to do far in advance of getting COVID-19. Some important things that these first 2 checklists will walk you through include looking up important phone numbers you might need to call ahead of time, having them in one centralized place, keeping a suggested list of supplies readily available, such as a pulse oximeter and a thermometer, for example, and how to find where exactly you're going to go to receive both testing and monoclonal antibody therapy. Please note, if you don't know off the top of your head exactly where you can immediately obtain monoclonal antibody therapy, within the directions, there's a link provided on item number 5 that will help you find the location nearest you. You simply click on that link, enter your zip code, and then a list of locations will appear that you can choose from. The next two checklists include what to do if you have known exposure to COVID-19 or a positive test result. These two documents will guide you through exactly when you should obtain a COVID-19 test after known exposure, how long to be monitoring for symptoms after exposure, and perhaps most importantly, how to adequately quarantine. If you aren't sure what steps you should take to try to keep from getting COVID-19, especially if someone else in your home has already tested positive, then this is a very valuable tool that you can use. Please read through this checklist so you can be well informed and be prepared. The last checklist I'm going to be sharing with you today is perhaps the most important because it tells you exactly what things to do after testing positive for COVID-19, which for some can be a very stressful time. This checklist reminds you of the steps to take early on, one of which is to call your healthcare provider immediately to discuss how you can receive monoclonal antibody therapy as soon as possible. It will also prompt you to keep a log of your vital signs and your symptoms, what to remember to do just in case you need to seek emergency medical care, and steps to take after your illness has run its course. You can find all of these checklists on our website at cllsociety.org. As you can see in the screenshot of our website, there is a COVID-19 update button located near the top of the page in red. And if you click on that button from the homepage, it will take you to our dedicated COVID-19 information page. Then in the middle top of the COVID-19 page, you will see a large icon that says COVID-19 Action Plan. After clicking on the action plan, you will find all of those checklists that I have reviewed with you today. So please remember, the COVID-19 Action Plan is there for you on our website whenever you need it. But we highly recommend that you access it ahead of time before you are exposed and before you start feeling ill. Doing so will help you have peace of mind, knowing exactly who to call, what to have on hand, and what to do in the moment. After you complete your plan, again, we suggest you keep your checklists in a folder, place them in an easily accessible place. It's also very important to discuss your plan with others in your household so they too can know what to expect. And please remember, if you have known exposure, any symptoms or a positive test result, act fast. Even if you don't yet have symptoms, call your healthcare provider right away because obtaining early treatment, such as monoclonal antibodies, are very important for achieving the best outcome possible, especially for those with CLL. Thank you, and please stay safe.
Thank you, Robin, for that succinct overview. I can't emphasize how important this is. We're going to move on from that, but we're going to circle back to it again. But let me start by introducing our speakers. And today we're joined by two CLL experts, a virologist and an infectious disease specialist, as Patty pointed out. Dr. Daniel Engel is helping us for the first time. He's a professor of microbiology, immunology, and cancer biology, as you can see, at the University of Virginia School of Medicine. And he's going to be talking a little about the biology and the different clades or different variants of the COVID virus. We also have Dr. Deborah Stephens, who's helped us before. She's an assistant professor of hematology and hematological malignancies at the University of Utah Health and at the Huntsman Cancer Institute. Nicole Lamanna is the associate professor of clinical medicine at the Division of Hematology Oncology at Columbia University in New York, New York. And we welcome back Dr. Sanjeet Dadwal, who's chief of infectious disease at the City of Hope's National Cancer Medical Institute or Center. This is the outline of how we're going to plan to do this, and hopefully we'll have most of the time spent in Q&A because we've received hundreds of questions, and I can guarantee we won't get to them all, but we'll try to get to as many as we can. And I think we're going to run through some polling questions next. So those should pop up on your screen. Question one is, have you contracted COVID? 2 is, have you received or plan to receive the COVID-19 vaccine? Have you received or you plan to get a third booster shot? Question 4, how protected do you feel by the COVID-19 vaccine? 5 is, do you plan to continue to mask indoors and socially distance? And 6, if it were to be authorized, would you use an anti-SARS-CoV-2 monoclonal antibody such as REGEN-COV or AZD742 as an ongoing pre-exposure, so before you're exposed to COVID, prophylaxis to prevent COVID infection? And here's the results here, and we do see that 5% of our audience has had COVID, that 98% have received the vaccine. I'm actually surprised there's 2% that haven't, but we can find out more about that. The booster, we have 91% have had the booster, but 7% are unsure, and there was lots of questions on that. So panelists, be prepared for that. And this is interesting. Only 3% of you feel very well protected, but at least 27% feel protected. And there's kind of a split here between neutral and feeling unprotected and very unprotected after the vaccine. Are people continuing to mask indoors? And 3 out of 4 you continue to always mask indoors, and the majority of the rest are often masking indoors post-vaccine. What we say is get vaccinated, but act as if you're not vaccinated. And let's see what the results are. So it looks like there's a lot of enthusiasm about uptake. Again, 3 out of 4 of you would consider the monoclonal antibodies. Very interesting data that I'm sure the panel will address. Thanks everyone for filling out that survey. All right, so we've seen the agenda, so I think it's time to get to our panel here. And we're going to start with Dr. Daniel Engel. And our question for you is, where do we stand with variants right now? And what does this mean for the immunocompromised CLL patient?
Good afternoon, everyone. It's an honor to be here. I'm speaking to you from my laboratory at the University of Virginia in Charlottesville. I want to address the question about variants by touching on a few topics that are listed in the first slide that you can see now. So how do these variants arise? As you probably heard, viral variants arise by mutation of the virus's genetic material. You may have heard on the news the phrase viruses mutate in talking about the variants. This is true. We know that the SARS-CoV-2 virus mutates constantly as it replicates in humans. This is a natural outcome of replication, and viruses in general do this all the time. Now, a little bit of virology here, but I think it's somewhat practical. The overwhelming majority of the time, these mutations do not alter the virus. The mutations are silent. However, extremely rarely, a mutation or a set of mutations will occur in the population that gives the virus a significant advantage over the original virus from which it came. And this allows the mutated virus, the variant, to infect more people and become predominant. So that's kind of how it works. What does this mean for the pandemic? It means that the more infections there are in people, the greater the chances there are for additional variants to arise, because more infections allow these extremely rare variants to be generated. Therefore, the fewer infections there are, the less likely there will be new variants. So the keys to preventing variants from coming up are the same as the keys to getting infections down. These are our public health measures: vaccination of the population, masking, social distancing, and hand washing. So will there be new variants? And is there anything that we should do to stay protected against new variants? So now you can understand why there will likely be more variants as long as there are people who are getting infected in super high numbers across the globe. Our means of protection are really the same as the tools that we have now, as I mentioned: vaccination, masking, social distancing, hand washing. So my colleagues on the panel will be discussing vaccination, we'll be discussing monoclonal antibodies, but specifically, I'll just briefly mention how the variants affect vaccination and monoclonal antibody therapy. As far as the variants are concerned, there's the possibility that both vaccination and monoclonal antibody therapy could become less effective as new variants arise, because the current vaccines and the current monoclonal antibody therapies were not designed against new variants. They were designed against the existing virus or the original virus. However, people are working extremely hard on designing new vaccines and new monoclonal antibodies in order to keep up with the changing landscape of new variants. And then finally, I want to address the issue of masking that's already come up and which all of you are familiar with. And the results of the poll, I think, are very good. So I just want to reinforce what you may have already heard. Based on the science, the best mask to wear is an N95 mask. And based on the science, I highly recommend wearing an N95. It provides the best individual protection. Cloth masks do not provide enough protection, and I recommend against them. Surgical masks are okay, but do not provide the highest level of protection. You've heard of KN95, which are produced in China. Those are very good, but they're not made to the same standards as N95 masks. N95 masks are only made in this country, and there are no questions about what source they come from. And you can get N95 masks on Amazon, and it's where I get mine. So I just wanted to mention that. And then lastly, on the next slide, I wanted to make you aware of a group that I'm involved with at the American Society for Virology. We run regular virtual town halls where anyone from the general public can register. They're free of charge, and you can ask questions of panel members about all aspects of COVID-19 pandemic and vaccination in particular. So I invite you to join us. Just go to asv.org/education for more information and to register. And I thank you, and I look forward to answering your questions later. Thank you.
Dr. Engel, thank you so much. That was really excellent. And I'm really echo what you say about the importance of vaccines and understanding how the virus evolves. We're lucky to have Dr. Deb Stephens join us again. On your question, Dr. Stephens, is what have we been finding out about vaccine response in CLL patients specifically, and how should this affect their behavior?
Thanks, Dr. Kaufman and the CLL Society for inviting me to speak today. I think this is a really important topic, and it's evidenced by the survey question that asked, do you feel protected by your COVID vaccine? And I'm going to talk a little bit about the data that's out there and my recommendations on what to do based upon these. You could go to the next slide, please. We really have most information about the Pfizer vaccine right now, just because it's the first one that came out, and it just takes a little while to get all these data published. So the first study that came out specifically for response in CLL patients was done in Israel. And that study detected responses by measuring antibodies to the COVID spike protein. And essentially the overall response rates were about 40% as based on antibody titers in CLL patients. They broke these down by what period of the CLL the patients were in. And patients who were in remission after therapy, about 79% of those did respond to the vaccine, which is a great number. However, patients even just on watch and wait, only about 55% responded, and patients on essentially any kind of treatment, only 16% achieved an antibody response. If you could go to the next slide, thanks. So then the group at MSK did put out some of their data about responses to the Pfizer and Moderna vaccine in CLL patients. Similarly, they were looking at antibody response, and they were able to detect response in about 52% of CLL patients. However, they found a little bit higher number of patients who were never treated at 94% that did achieve a response by antibody. Patients with any prior therapy was quite low response by antibody, 23%. And I broke down a couple of the specific drugs that they studied, because I know several of you probably know about these drugs or maybe on these drugs currently. So the drugs ibrutinib and acalabrutinib, about 21% of patients were able to achieve a response to the vaccine. Anybody who had received the CD20 monoclonal antibodies, rituximab and obinutuzumab, within the last 12 months, only about a 14% response. Anybody who received those antibodies plus the pill venetoclax within the last 12 months, 0% of those patients responded. In this particular study, they found patients who are younger, and fair enough, they defined that as less than 70 years of age or no prior treatment were the ones who were more likely to develop antibodies. We go to the next slide. So I just put this cartoon up to describe different types of antibody response or different types of vaccine response and where the antibody response comes in. So as you can see by this graphic, when a patient gets a vaccine, there are different parts of the immune system that might respond to that vaccine. And so you can see here up at the top, this is a cell called a dendritic cell. There's also one called macrophages, which take the vaccine particles into the immune system and allow the T cells and B cells to respond. There are things over on the right-hand side called cytotoxic T cells or plasma cells, and then memory B or T cells. So these are all parts of the immune system that might respond to the vaccine. However, the antibody response is the easiest one to test in the blood. And so that's why all of our studies so far really have commented on what is the antibody response. And so you can see that there actually may be additional responses besides antibody response. So you could go to the next slide. So again, because there are likely responses outside of the antibodies that are not easily measurable, and at this point, the risks of severe complications of a COVID-19 infection outweigh any risks of the vaccination. We do still recommend that all CLL patients receive a COVID-19 vaccine. At this time, the FDA has authorized, if you originally received a Pfizer or Moderna shot, you can receive a booster shot and it is recommended that you do the shot of the same vaccine you originally received. And I know a lot of patients have asked me, well, you know, we have checked my antibody response and, you know, maybe I got the Pfizer vaccine and I didn't have a response to that. Should I then get Moderna or J&J, you know, switch classes of drugs? And actually, you know, it's still early because these booster doses just were approved, but I have seen patients that didn't make a response to their original 2 doses, then got the same dose and did make antibody responses to the 3rd booster dose. However, even if we can measure antibodies, we don't have great data to say exactly how many antibodies are needed to protect you fully against infection or severe infection with COVID-19 infection. And so just as, you know, Dr. Engel just mentioned, I really recommend continuing to be vigilant with masking, washing your hands, and, you know, social distancing as appropriate. I know this gets harder and harder because a lot of patients were expecting these vaccines to be, you know, their get out of free get out of jail free cards. And then with, a little bit question in the vaccine response, it just becomes longer and longer that patients are practicing social distancing. And so, you just have to keep in mind the risks and benefits and make sure you're not isolating yourself either. And this is my last slide on this. So thanks for your attention.
Thank you, Dr. Stephens. That was a nice summation, and Dr. Lamanna is going to answer a more difficult question here. They're all difficult because we don't have the answers. This is a moving target, isn't it? If a CLL patient becomes infected with COVID, what are the immediate next steps? What treatment should they be asking for? And you were in New York at the epicenter of this a year ago, but I think it's less of a pressing issue now, but tell us what you're recommending to your patients, because you and I have had several offline discussions about what you've seen as a flood of patients, and now, thank God, it's more of a trickle of patients.
Yeah, thank you so much for having me. This is always, I think, obviously the biggest topic I think that probably Deb and I deal with in our clinic much of the time, despite, also conversations about therapies and so on and so forth. And obviously we understand how pressing this is for all of you all. And as I said, it's an evolution. So clearly I want to thank CLL Society, Brian and Patty, for doing these kind of forums, because things constantly change. And I think you all need to know that, is that things are constantly evolving, and medicine and science are constantly evolving, and therapies are constantly evolving. So that's the good news. But with that, I know there's a lot of unanswered questions. And so just got to take it for that. We're all in this together and rolling with it. But I think it's really important. So, if you do become infected, first is please stay calm. There's a lot more that we have now than we did last year. And what Robin developed with CLL Society together with Brian, the COVID plan, excellent checklist. So print it out, have it available, go through it. And many of those answers you might not know right off the bat. And that's the point, is that then you can contact your physician. And whether that be, in my case, I always have my patients call me for everything. But it could be your general practitioner, internal medicine physician, healthcare provider team, whoever you're aligning with very closely might have some of those, hopefully will have those answers on the checklist. And obviously they included links that will help you also find out where the monoclonal antibody therapies might be available in your area. So again, I think it's a very, very helpful checklist to go through and start getting those answers, so that if you should become infected, you stay calm, you have a plan, sort of, what to go through in a calm fashion and what to anticipate. With that, I think one of the take-home points is that now that we're going to talk a little about the monoclonal antibody therapies, and these are just some of the companies that are making monoclonal antibody therapies, and there likely will be more. But you want to make arrangements to receive monoclonal antibody therapy in a rather expedient time. So really, ideally, the guidelines are less than 10 days, but honestly, we really want our patients to receive this in a matter of a few days. And so the earlier you do this, I'm going to explain how the monoclonal antibodies work, but the earlier you do this, we're trying to shut down the virus and obviously shut down the severity of an illness within a person, an individual person. And I think it's important that there are two ways to receive the monoclonal antibody treatments, at least most of them. For Regeneron and Eli Lilly, they're approved in two fashions. And again, this is evolving, so this will change over time with more monoclonal antibody therapies. But one is if you have a positive test. So if you yourself have a positive PCR test, then you can receive the monoclonal antibody treatment. And then the other, which is less known, is that if you've had a recent been exposed to somebody, so your spouse, significant other, your kids, your grandkids, if they've have a positive test and you've been with them for a prolonged period of time. Now, I understand that's a little bit nebulous, but a significant time. I think the exposure they're calling is 15 minutes or more. I'm being facetious, but the point is, if you're with them for a few hours, somebody who was then identified as having COVID, a post-exposure prophylaxis, you yourself can also receive a monoclonal antibody treatment. So I think that's important to know. And again, I ally you to the fact that if you are not sure, you can call your provider. I think that's important. If your provider doesn't know, I'm hoping your CLL doctor does. So, you need to get answers, but hopefully this will get easier over time. Again, this is evolving and new. And then, of course, depending upon who's in your household, you want those individuals to obviously get tested as well and quarantine as well. And obviously the goal of this, and to also tell anybody that you might have been with that you recently have had a positive test, because the goal is to obviously try to spread other individuals from getting sick and spreading it to other individuals, so on and so forth. Next slide, please. So how do monoclonal antibody treatments work? And there are lots of different monoclonal antibodies, including the fact that many of you think about rituximab or obinutuzumab. Many of you might be familiar with those monoclonal antibodies. And in those sense, and those are different monoclonal antibodies, they're designed to attach antigen on cancer cells. And those in particular to attach a protein CD20 on your CLL cells. These are different monoclonal antibodies, and there are many. But essentially what these were designed to do was that these were designed to sort of neutralize the virus. They're made to attach itself to the virus. Specifically, the virus, what it does is it has a receptor binding domain that can attach to our cells and then sort of invade our cells. And that's how it sort of, and obviously Dr. Engel can do this much better than I can. But the goal is that obviously it replicates and it gets into the host cells, our cells, and continues to divide. And the more virus there is, obviously, the infectivity and severity of the illness can worsen. And so the goal of these monoclonal antibodies is to shut that down, prevent the virus from attaching to our cells, getting into our cells, and sort of neutralize the virus, and then stop it from replicating. So that will decrease the severity of an illness within an individual. So getting it early, and that's what we're talking about, will do that sooner. If somebody has had the virus for a long, prolonged period of time, a week, the virus has replicated a lot by that time, and the illness, has already, spread within that individual, and the symptoms might be severe at that point. So the goal is to get the monoclonal antibody treatment early. And there were studies even in patients who had severe illness. So this is really approved for mild to moderate symptoms. For patients who had severe illness already in the hospital, this didn't really help folks that much. And so that's the reason why this is an outpatient therapy that individuals can receive. Next slide. So as I said, the FDA has approved these monoclonal antibodies under what they call emergency use authorization, or EUA. So mild to moderate patients, adult or pediatrics, who has a positive test can receive this. Or the second that I just had spoken about was post-exposure prophylaxis. So somebody who has been in contact with somebody who's and is not fully vaccinated, and or has an immune compromised condition such as CLL, so not expected to mount a full response to vaccination. Those are individuals eligible to receive in post-exposure prophylaxis. So these are the two currently recommended, or I should say approved, usage of the monoclonal antibody therapies. But obviously we're going to be looking at different ways to use these going forward. Next slide. So just for those of you, and I don't know if anybody has received any of the monoclonal antibody therapies yet, but for those of you who haven't, it's typically given as an intravenous infusion, and it's about an hour each antibody, depending upon, again, there are several that are available and more to come. They're typically about a half-hour infusion, but they're going to monitor you as well. So you might be there depending upon where you get the infusion, whether it be at the hospital or in an office suite. They may monitor you for an hour post the infusion as well. So you might be there for a few hours, but the infusion itself is not long. Some can also be given subcutaneously as well. They're generally well tolerated so far from what we're seeing from the data that's available, from my experience as well. but you can have infusion-related reactions that could occur, and that's why they monitor you. So some people can have fever or chills. Some people can feel that they're short of breath or have palpitations. So these are typical of some of the monoclonal antibodies. Again, some of you who are familiar, different monoclonal antibodies, but with rituximab, we can see infusion reactions with that medicine as well. So they're going to monitor you, but they're generally, well speaking, very well tolerated.
Dr. Lamanna, thank you so much. I think this is a very hot topic, vaccines and monoclonal antibodies. Dr. Dadwal, you're the infectious disease specialist, and your sub area of specialty is dealing with the immunocompromised, working at a cancer institution. So you see a lot of patients that are immune suppressed who unfortunately have contracted COVID. So when a CLL patient is hospitalized with COVID, give us a sense of what we should expect, because most of us, we're now we really are nervous. We're going in a hospital. So tell us what we can expect, what it's going to be like for us.
Good morning, everyone. Thank you, Brian, for inviting me to speak here. And it's good to be back again. So I'll talk about patients who get hospitalized with COVID-19 who have CLL. Although, the treatment recommendations which are out there is mostly for general public, there's lack of data and specific recommendations in immunocompromised patients. I would say even in the IDSA guidelines, there's not much guidance as to use of antivirals in immunosuppressed patients, as they are more strongly provided for general public. And one of the major reasons is there's not a whole lot of clinical trial data in this population. And the treatment currently is based upon disease severity. The guidelines indicate mild to moderate. Typically, patients are outpatient. And as Dr. Lamanna said, they should be, sort of counseled to get monoclonal antibodies so it doesn't progress to the point where patients get hospitalized. I mean, this 81% reduction in hospitalization if monoclonal antibodies are given in time. So patients who get admitted are typically those who have moderate disease or severe disease, and there are definitions of these particular situations. Moderate is if a patient has fever, has pneumonia, but they're not requiring oxygen, that's moderate disease. And sometimes that can be managed as an outpatient too. And severe disease is when someone is requiring oxygen or they have oxygen saturation less than 94% on room air, meaning without any supplemental oxygen given. And critical illness is when patients are being transferred to ICU. Next slide, please. So the currently available antiviral drug is still the same what we had last year. Nothing has changed there. We have remdesivir. It's the only FDA-approved antiviral drug. It did not show sort of mortality benefit, but it did reduce the length of stay and severity of illness. And there are some ongoing studies, especially molnupiravir, which is made by one of the US companies. It's a phase 3 trial. And the intent is to treat those who are not hospitalized and to prevent progression to severe disease. Remdesivir is indicated for patients who have severe illness or moderately severe illness. And immunocompromised patients can benefit from that, although the data is not out there. Now, the other thing is anti-inflammatory approach. This is reserved for patients who are severely ill or critically ill, and they require supplemental oxygen. One of them is dexamethasone, which is a steroid. Now, one of the main things is the steroid should only be given if a patient is requiring oxygen. If they have pneumonia, but they are not requiring oxygen, it should not be given because it can increase the risk of fungal infections. IL-6 inhibitors. IL-6 is a cytokine which causes a tremendous amount of inflammation. So there are certain drugs which have been recommended to be given. One of them is tocilizumab, which is currently in a severe shortage around the country. And the alternatives at this time is sarilumab or baricitinib. These are the drugs which can reduce the inflammatory response. And the recommendation for giving tocilizumab or the other drug is when the patient is going from severe to critical, meaning if they're going to be transferred to ICU, if that is instituted within 24 hours of going to ICU, it has been shown that it may reduce the severity of the illness. Next slide, please. Monoclonal antibodies, this has already been mentioned by Dr. Lamanna. very nicely, so I don't think so I have to waste anybody's time. However, I would like to say this, when it comes to immunocompromised patients, one size fits all approach does not help. It has to be really individualized for each patient. A CLL patient who is on observation without therapy is different than CLL patient who is getting, let's say, ibrutinib or getting rituximab or certain other drugs. They all have different risk for developing severe disease or progression. So it has to be tailored to each patient. So at my institution, if we have a CLL patient, myself or one of my colleagues get a call, we look at the whole detail. If it is outpatient, what to do. If they require hospitalization, what do we do? We have a very low threshold to start remdesivir, even if they're not on oxygen, depending upon what their immunologic status is. And the other thing is, which can be done on an investigational site. So if you have clinical trials at your institution beyond remdesivir, do you have access to those, or there are local institutions where you could be, have that access? And lastly, the monoclonal antibodies, they're not recommended to be given in severe disease because it's mostly an inflammatory path. However, there are case reports and there are proposals to use monoclonal antibody in somebody who is shedding for a prolonged period of time. And CLL patients, as well as lymphoma patients, can shed the virus for a prolonged period of time. The other one is convalescent plasma. Again, I'm saying most of the data is in mild to moderate disease to prevent hospitalization. However, there are some studies which show that there could be benefit in treating immunosuppressed patients, even if they have moderate to severe disease. Next slide, please. And I think the most important thing is to assess, whether your cancer center or your local institution is sort of looking at each patient individually as to what their risk is for disease progression, and then act accordingly. And always ask your treating MD, I always say your CLL physician, to guide management. And then clinicaltrials.gov, you can always explore if there are any clinical trials ongoing. Thank you.
Dr. Dadwal, thank you so much. So now is the time for questions. And between the questions that people sent in before and the questions received after, we're going to not get through them all. I am going to say this, if you have a question that we don't get to, you can send it in to our Ask the Expert, and we'll try to answer it. And I'm not going to answer the specific CLL questions people asked about specific CLL topics. I'm going to just try to keep it to COVID on this, so I apologize in advance for that. Let me start with this and with the question on masks, and there was a number of these that came up. And let me make sure, Dr. Engels, there you are. You talked about masks. Can you tell us, there was questions, can we reuse an N95 mask? Can we reuse a KN95 mask? People are worried about counterfeit masks. What are you telling your friends, the patients, the doctors that consult you about mask use?
Yeah, you certainly can reuse the N95 masks. We don't really know how long that they last. That's kind of an open question. The reason that they're considered to be not reusable, quote unquote, is because when physicians wear those masks, and those are the masks that physicians wear when they're interacting with COVID patients, they don't want to use the same mask for two different patients. So they're considered to be not reusable in that sense. But it's okay for an individual to continue to wear an N95 mask. The sourcing of the N95 masks is really not questionable. They're all what's called NIOSH masks, and so they've undergone quality control and they've met American standards for the N95, and they're only produced in this country. The KN95 masks, I have personally seen variability in their qualities. the ones that I've seen that seem the best, and this is just based on how much air that you can push through them, are the ones that are distributed by companies in the United States. All KN95 masks come from China, but we don't always know, what their source is. So if you're going to wear a KN95, I would recommend that you get it from an American distributor, because those seem to be, and this is just my kind of casual observation, they seem to be more reliable.
And just one follow-up question, test fitting, that seemed to be big at the beginning. Your N95 should be test fitted. We're not hearing as much about that. Can you comment on that?
Yeah, my impression is that so in the beginning, people were being dissuaded from using N95s for a good reason, which was there was a shortage, and it really was something that the physicians and healthcare workers should have first dibs on. And so as part of that, people were reminded that they really have to fit right in order to work. Well, it's pretty easy to tell if you're wearing an N95 mask properly. It simply has to have, contact around your face, and you have to press in the nose clip so that there's a good connection between the mask and the top of your nose. And that's pretty much it. Remember, you're comparing that with a different kind of mask. You're not really comparing that with what happens in the hospital, as Dr. Lamanna might be able to tell us about. So my recommendation would be not to worry about official mask fitting, but to just make sure that there's a good apparent seal over your face, and then it's not loose and floppy. The only other thing I'll say about N95s is that they don't fit over your ears. They fit behind your head, which is inconvenient. So they're better masks, but they're harder to get on and off, and sometimes the straps will break. So it's less convenient, but it's a better mask, and you have to decide what it is that you want to put up with. If I get on an airplane, I'm wearing an N95, because I find the KN95s or anything that fit over my ears just to get really uncomfortable after a while. Maybe it's my ears. So that's what I do personally.
Does anybody else want to add anything on masks? Dr. Stephens, Lamanna, Dadwal, or did Dr. Engels nail it?
I really think Dr. Engel nailed it, but I think, generally I would just say any mask is better than no mask at all. So, whatever you have access to, make sure that you have a mask that has reasonable protection for you. If you can't get an N95, then, use whatever you have available.
And the surgical masks protect others. And when the community was wearing surgical masks, ironically, I think we needed the N95s less. I think we kind of need them more now. But fortunately, they're available now, and you can get them a lot less expensively. And I'm recommending to everyone who's listening, order them now, get them now, because maybe in December, they'll be hard to get, and they're also protect against wildfire smoke and things like that, which the other masks don't do, which is an issue in California and other states and stuff. So I think having them around is just a good idea. While we're on this, people have asked in this, again, I'll send this one to you, Daniel, air purifiers, HEPA filters. People have put these, you see these in restaurants, people have them in their homes, they're in a lot of hospitals. Do they make sense? Talk to me about that a little bit.
Well, HEPA filters certainly do good in filtering out microbes that we're sure about. It doesn't protect you if you're standing next to someone who is shedding virus and is not masked and is at the peak of their illness. it does not filter that air for you. So you can go into a HEPA filtered room and have an encounter that could be dangerous. But in general, being in a HEPA filtered room is a good idea, because if there's someone who came into the room or is further away from you, et cetera, that air is going to be moved and will be filtered.
Doctors, go ahead.
And I may add to that is also the ventilation of that room. And so you have HEPA filter, but is there air being circulated as well? So that's the other element as well. So if you're in a closed space, like a small restaurant, even if you put a HEPA filter, it may not help in sort of reducing the risk of acquisition of the virus if somebody is out there.
And before we move into some more active therapies, I just want to talk about the social distancing. And we're hearing a lot less about food. And, you know, we were at the beginning, we were washing every bag that we got and letting the mail sit for 3 days. Is all of that stuff, the transmission, the concerns about transmission. I'll ask you this, Dr. Dadwal, that, you know, through food, through picking up an envelope, you know, through pumping gas, things like that, that people wearing gloves and hand sanitizing pre, post, after. Tell us where all of that is at now, because I think we have a lot more science on that.
Yeah, I mean, folks were really, really concerned about fomites as a source of transmission by touching onto things. But I think that has been sort of, doesn't seem to be the major route of transmission. It is still the air. I'm saying you're close to somebody who's breathing in your face or coughing or sneezing. That's the main thing. But in a way, I think it is good hygiene. If you are touching trying to fuel your car and you don't know who came before you in touch, because there are other viruses like RSV or influenza which are really fomite driven and which could be equally bad in immunocompromised patients. So I would say when multiple people touch and you touch, it's a good measure, just hand hygiene, have that alcohol, clean your hands, wash your hands all the time. But we don't have to be that crazy as we used to be, like you said, let the mail sit for 3 days or wipe down everything, but it still may not be a bad idea to keep your things clean. But it's no longer felt that that's the major route of transmission.
Thank you. Dr. Stephens, the vaccine, one of the controversies, and you gave the official line, if you got Pfizer, you get Pfizer again. And if you got Moderna, you got Moderna again. But people are reading that the Moderna vaccine is better in the immunocompromised. It has 3 times the amount of viral protein in it. People get stronger antibody responses to it. And not all doctors are saying what you're saying. Tell us a little bit about your thoughts on that. Is it wrong for somebody who's had the Pfizer maybe to switch to the Moderna or to switch to something else? What are your thoughts on that? Because I know there's not a lot of science behind it, but there is definitely some evidence that the Moderna is a more potent vaccine.
Yeah, and I think it's just we don't have enough formal scientific data to make strong recommendations either way. When the FDA emergency access use came out, they did recommend that you use the same vaccine that you had your initial response to. But really, I think there are lots of studies ongoing right now that are looking at, what happens if you get the Pfizer vaccine first and then switch to Moderna, or the opposite way, and different combinations. And I think once there's more data, we'll all feel a lot more comfortable making recommendations. I personally feel like getting the 3rd booster dose is pretty important for CLL patients. And if it's not possible to get the same vaccine that you got previously, I would recommend just, getting whatever one you have easy access to. And I mean, thankfully here in the United States, at least, there are pretty wide access to vaccines right now. And especially without, they just authorized 65 and older patients to get the third vaccine. So I don't know if that will affect our vaccine supply at all, but I think right now we're in a good spot to get whatever vaccine is available.
So following up on that booster, and I think all of us would recommend a booster for all CLL patients, whatever stage they're at. But are there some people who shouldn't? And I'll give some questions that were here. Someone had autoimmune hemolytic anemia after the second dose of the vaccine. Someone had a very painful lymph nodes and their spleen enlarged. I've also heard of people's ALC, their absolute lymphocyte count, shooting up after the vaccine. So what are you telling? And then other people who are just miserable. I mean, flu-like, miserable for 3 days, and then they're better. What are you telling those people about getting the booster? Dr. Stephens, you want to take that?
Yeah, and this is actually fairly common for some sort of reaction to the vaccines. And, you know, I think if anybody had a really severe reaction to one of the vaccines, you know, severe skin rashes or allergic type reactions, they should be, you know, very cautious or not get the same vaccine that they got initially. The lymph node responses are very common, and I actually warn people about them before they get their vaccine because, of course, in a patient who has CLL, it's pretty concerning to see a lymph node pop up really quickly. I mean, people are thinking, is my CLL progressing? Am I transforming into more aggressive lymphoma like Richter's? But it's fairly common in patients with CLL and without CLL. And the thing to remember is that even though likely majority of your lymph nodes are containing CLL cells, there are some normal immune cells in there, and their job is to respond to any sort of insult. And, you know, getting a vaccine is like, you know, testing your body to see how they can respond to this virus. So, you know, your body may respond as if you have an infection, and that's where the fevers, you know, fatigue, you know, enlarged lymph nodes, all of those things happen. And I know that it is very inconvenient to go through these side effects, but I would assure you that it is more inconvenient to have COVID and to be hospitalized. And so I'm really, you know, especially those reactions that look like more immune reactions that are just, you know, the immune system responding to the vaccine, I really do encourage people to go ahead and get the vaccine again and just be prepared. You know, let your employer know you might need a couple of days to recover from this, but it's way better than actually getting COVID and needing like weeks off of work. And so, you know, although inconvenient, you know, some of those side effects just indicate to us that the body is, you know, responding to that vaccine.
We know all vaccines are associated with autoimmune problems. And I just wonder if you could comment on that. And Dr. Lamanna, I don't know if you want to take that. And I know you had something else to say. Go ahead.
Yeah, no, I just wanted to echo what Dr. Stephens was saying is, I think if somebody, because I read some of the, for sure, if somebody's had a severe reaction to the vaccine, I think it's important to talk to your provider about whether or not you should, you know, receive another vaccine or what to do in that scenario. So it is not to say, millions thankfully have tolerated these vaccines very well, but it's not to say that we haven't had anybody who's had some toxicity or adverse issue come up after receiving their vaccine. So we do appreciate that. We're not trying to minimize that. I think obviously, given the devastation of COVID, we're trying to balance all these issues. But the immune mediated reactions, the lymph node swelling, redness, fever, chills, feeling bad for a few days usually is very short limited. And, you know, I think that what Dr. Stephens is trying to say is that is, you know, you'll get through that part. You might be miserable, but that's more important than obviously not receiving a vaccine and getting COVID. So I think there's a severity of symptoms that are more expected. And the lymph nodes can take some time to calm down. And we can even see that with other vaccines, not just the COVID vaccine. So it's important to know that. But I just wanted to say that. And then the autoimmune issues, absolutely. And I think that there was somebody who commented about having autoimmune hemolytic anemia or something after they received the vaccine and they got treatment and so on and so forth. And I think that's an important thing as well. So obviously you need to stay in close, with your provider, because there may be an occasional autoimmune issue that can occur because of your immune dysregulation that might be different than somebody else. And so we might have to interact on that behalf, because sometimes viruses and infections can actually set off the autoimmune process. And so that can happen, forget COVID vaccine, that can happen to CLL patients where something has set off their autoimmune cascade, autoimmune hemolytic anemia or thrombocytopenia. So it can happen absolutely for other reasons as well. So then you might need to be treatment for the autoimmune complication that happens. So
Are you recommending the family members get the booster shot to a spouse or someone to protect their family member? What are you saying in those cases, Dr. Lamanna?
Well, thankfully, hot off the presses, as Dr. Steven just noted, it just got approved for 65 and older. I mean, obviously, yes, if we have access to that, of course, we would love that the close family members of somebody who's got an immune-mediated condition get the vaccine, but we understand there were barriers to that. Hopefully that will change given the recent, approval, that it'll be much easier to obtain that. And I think it's not just for 65. I think the caveat they listed was even in folks who might be inherently at risk all the time, and that'd be healthcare workers who are constantly in contact or exposed in a different way with individuals who might have COVID. So I think that the criteria is loosened up. So I think it'll be more available, and hopefully people will take advantage of that.
So there's a whole bunch of questions on whether the vaccine is going to work. First, and this is controversial because I've seen results on both sides. If I have a horrible reaction or I have no reaction, does that tell me anything about how much protection I have from the vaccine? We like to say, oh, that sore arm, that fever, that means the vaccine is working. But is that just kind of whistling in the wind? Does it really mean anything? Do we know? Dr. Lamanna, you want to take that, or anyone want to take that?
I'm happy to take that.
Go ahead.
I'm sure we're all happy to take that.
Okay.
I think, I mean, obviously, the some of the data that was presented, and it's evolving, about the variability of immune response in CLL patients and other immunocompromised patients. Obviously, I think the end result, as we get the science to catch up and the data to catch up with what's going on, I think the proof in the real pudding, so to speak, will be on what we see in hospitalization rates and mortality. And I'm going to tell you, as somebody who lived through this in New York, being one of the first epicenters last year, this year in New York is far different than what it was last year. And I think in the end, as we gather, as I said, all the immune responses and whether spike antibody testing is the right antibody testing to do, because we know that there's probably, as Dr. Stephens beautifully showed, there are other cells that also help with your immunity that we can't capture, and we're learning about and how to figure that out and what testing might be provided. I'm seeing far less of my CLL patients hospitalized this year who are vaccinated, meaning people doing well, than I did last year. Completely night and day difference. And so I do think that the vaccine has provided benefit, and obviously we'll be teasing through that data about those individuals. And we know that treatments can affect vaccine immune response and so on and so forth. But I think that it is still far better this year than it was last year, depending upon the region and what you're going through. And again, we will see how that evolves over time, and hopefully we'll be able to provide more meaningful data about how to mount immune response. But I think that's why you're still hearing many of us urge you to get your booster shots, because we know that this is a constantly evolving situation as we go forward. And I'm sure the others can comment as well.
And we've had the same experience in Los Angeles too. I mean, at our institution, with this last surge we had, particularly LA County had big numbers. We probably had one patient with CLL who got hospitalized. That's it. And that person did fine too. So to Dr. Lamanna's point, I think it's very important to recognize that vaccinations, I think they have definitely helped in reducing the risk of hospitalization or severe disease. But one other bigger thing is to have every family member around you or friend you interact with be vaccinated as well. I mean, the issue of booster and family members is going to be sort of contingent upon what CDC, ACIP states, but they should be vaccinated. I think that's the best way.
Yeah, and another comment I'll make about kind of immune responses, based on people's symptoms. I don't think it correlates very well, but one thing I've noticed is when I'm checking antibody responses in my CLL patients, somebody comes in and tells me, I've got this big lymph node, I had fevers, for a couple of days after the vaccine, and then we run their antibody titers and there's no response by antibody. And so that's why I really wanted to bring up the point that there are likely other responses that we're not quantifying. And so, I don't want people to get the impression because they don't have an antibody response that the vaccine has not had any effect on them. It's just that's the easiest thing we have to measure. And hopefully those tests that help us measure other portions of the immune system will be coming forward.
Let me stick my neck out here. Go ahead, Dr. Engels. Yeah.
Well, the other point is that from the original clinical trials, which of course didn't necessarily include immunocompromised people, so it's really from the general population. But with that as a caveat, in those studies, there wasn't a strong correlation between getting a sore arm or fever or reactogenic kind of things with the effects of the vaccine, because those, in the beginning, we saw that the mRNA vaccines were up to like 95% effective in preventing hospitalization and death, where 95% of people don't get a fever. It's lower than that. So therefore, it's possible, at least in the general population, I can't extrapolate to the immunocompromised population, but in the general population, you can get a no or a mild reaction to the vaccine and still have outstanding protection.
So let me deal with this antibody question because it's come up in a whole bunch of different ways. And what I'm hearing you say, I think the panelists would all agree that if you have a zero response on antibody levels, which is really the only thing we can commercially measure, there are research labs that can look at other things, and there are some very esoteric people looking into neutralizing antibodies, and there are some T cell functions out there that people can order and stuff. So there are some things that can be done, but generally, commercially, it's a spike protein antibody. If people have no response, that doesn't mean that they don't have that they didn't respond, that they still may have some protection. And if people have very high levels, like after a booster, it's over 2,500 on the LabCorp test, let's say, that doesn't mean that they can, as one of our group leaders say, go around licking doorknobs and jumping in mosh pits. They're not completely protected either. I think that's true, but isn't it also true that if your levels are really high, you may have some level of protection? It may be the opportunity to hug your grandkid again, even though your grandchild hasn't had a vaccine, especially if you're in a community like LA or something, or New York now, where the levels are pretty low, and not if the grandkid has sniffles or they've been exposed, obviously not. So are you using that? How are you using it? Or you just say, forget it, don't bother to do it? I know this is a controversial area, but there's a ton of questions about this. So I'd like to put, the docs on the spot in terms of that. And maybe I can get just a quick roundtable of everybody on that. Yeah.
So, we actually do not recommend doing antibody testing on a routine basis because there's really no defined threshold where we can say you are protected or not protected. So that information really doesn't exist. Although I'm saying if you have an antibody response, all we can say is you responded to the vaccine and hope that it is going to help, right? But we don't know if the level is 500, is it better than 200? Is it better than 100 or 1,000 is better? That we do not know. So I think that is the reason why CDC still doesn't recommend doing antibody testing. that's their recommendation, although it is commercially available if somebody wants to know. And especially I want to highlight that if a patient who is actively getting anti-B cell therapy, let's say anti-CD20 antibodies or otherwise, we know they do not respond very well. And also you need both B and T cells to function together. So if one arm is completely sort of, unavailable due to the therapies for B-cell lymphoid malignancies, then I would be a little careful in assuming that perhaps you have a good T-cell response also.
Fair enough. I agree. I think that it's important that what you started off, Brian, saying earlier is, pretend like you haven't been vaccinated, even if you have. There are extremes. We're obviously understand how difficult this is for everybody. But I tell my folks, I am obviously checking antibodies, but more of it is for academic reasons at this point, right? To try to understand better in different patient populations within CLL on different therapies, and also serially to try to figure out, where serial testing, because maybe you may have 250 for one moment and then, 0 the next moment. So we really, this is a gathering. Remember, this is the first year the vaccines have been available. We're learning about what's going on, and we're learning and gathering this data. But I agree, you shouldn't falsely rely on your antibody levels. So but you want to make some practical sense of life, and we understand that. So you're trying to, I think that if there's, the nice thing about the differences between when this first started, testing's more readily available. The fact that people who have normal immune systems, remember the vaccine, I think one of the issues that people always ask about between vaccines and the monoclonal antibody treatments are, if the vaccines don't prevent infection, which they do, then we should just get the antibody therapy. And those are very different things. So I think it's important to know that if those who have healthy immune systems and your family and friends are getting vaccinated, yes, it's not to say that you can't get breakthrough infections, but the point is most of those people are doing well and at home. And if they have symptoms, sure. So if you have a loved one who's been vaccinated and has symptoms of a cold, you might not want to be around them. But if you're somebody who's a loved one who's been vaccinated and is otherwise well, then, it doesn't mean that you can't see them or be with them. So you have to make some prudence of this. And if there's a concern where you develop some symptoms and you're not sure, the testing is readily available. So that's something that's very different than last year. And as I said, we're seeing that patients in general are doing so much better than they were last year that I have not had thankfully, anybody really admitted with CLL and COVID in a really, really long time since earlier this year. And that's a huge difference than what I went through last year, rounding on COVID all the time. So I think that there is some assurances. I'm not trying to say don't let your guard down, but you have to make some sense of this as we're evolving this together. And so you need to use some practicality. So don't rely on the antibody levels. Of course, we're more reassured that if you've gotten an antibody level, certainly it means probably something versus if you have none. We agree with that. And the fact is that you need more than just your T cells and other cells that have an impact along with your B cells. And so we understand that there's a variety of different immunity here, and we don't understand the robustness, and it will be individualized depending upon each of our CLL patients and what they're going through. So you need to take a deep breath, and as we evolve the data and make practical considerations for your life, but know that there's a lot more we can do this year than we can last year.
Yeah, I would definitely echo what Dr. Lamanna just said. And I think one thing, is just thinking about being reasonable. And I think everything is about risks and benefits. And, there's nothing, it was a little bit easier to say, you can't see your grandkids last year when we weren't entirely sure how long this whole, pandemic was going to last. And as it gets longer and longer with continued risks, I fully realize, some of my patients are quite elderly, and, just balancing that, in reality, they may not have that many years of life left. And, maybe the benefit of seeing your grandkids, it outweighs any potential risk. And so again, if these, if your grandkids are healthy, if they're old enough to get vaccinated, that's an even safer situation. But there may be situations where it just makes sense from a risk to benefit standpoint that, it makes sense from a mental health standpoint, or, you can take precautions as much as you can with masking and having them mask and making sure everyone's using hand hygiene and creating as much physical space between you that it just might make sense for you to be able to see your families again.
And plus there's, like Dr. Lamanna said, there's very good access to antigen testing, which can be done in 15 minutes. And if you're negative that day, you visit, you're fine. So yes, it's important to have good mental health. I say it's very, very critical, and it has been very disturbing for everybody. I'm saying personally in my family, my in-laws, they're in their 70s and they're on the East Coast, and I have an 11-year-old and he cannot fly because he's not vaccinated, because he's worried he's going to give it to them. So, and if you bring in the element of being immunocompromised, that's even scary. But like you said, this year it's better than last year. We have more things up our sleeve which we can use and mitigate the risk.
I'm going to switch topics here. Just one last question. I can get some quick because we're running out of time. What are you doing, Dr. Stephens, about BTK inhibitors? Are you holding them? I think the NIH is doing a trial now. That's a question that's come up a lot. And if there's other therapies where you're thinking, I know that we don't have any data. Are you doing anything practical? Because the data on people getting the vaccines on BTK inhibitors is disappointing. Yeah.
Yeah, so there's a lot of things that really have to be personalized to the individual patient when talking about this. So I wouldn't say anything is a good blanket statement for this, and I would definitely talk to your cancer doctor about any specific recommendations that they have for you. But in terms of if somebody gets diagnosed with COVID that's on a BTK inhibitor, if they don't have a severe infection, meaning they're not in the hospital, they're not on oxygen, I typically have them continue their BTK inhibitor. If they have a severe infection where, of course, they need mechanical breathing devices placed or anything like that, I'm holding those therapies. So again, I think it's really individualized to where the person is at in their CLL treatment, because maybe, you've been on ibrutinib for 5 years and you're in a really great remission, and it might make sense to hold that drug to give you some more chance to recover, but it's really pretty individualized at this point. As far as responses to COVID vaccines, I have not requested that anybody hold their ibrutinib or acalabrutinib in order to gain a response, because I still think the benefit of treating the CLL and maintaining that treatment outweighs risks of stopping it and maybe your CLL getting worse, and especially in patients that maybe have been through multiple lines of therapy and they don't have a lot of good options after that treatment fails. And so again, I just think it's really very individualized. And so I make sure that you're consulting with your CLL doctor about specific recommendations for you.
Maybe somebody can take this one just quickly. IVIG, I know that some of it has anti-COVID antibodies in it. Is there any role for it in prevention, treatment? Any thoughts on that?
Yeah, I mean, obviously we don't know. I mean, I guess the end result is we don't know. I think that during COVID, depending upon where you were in the circumstances, last year, for instance, when I really wanted to keep my otherwise okay folks out of the center, we would hold things like IVIG just because I really didn't want them to be exposed by coming to the infusion center. Switching to home IVIG or subcutaneous, something where they were not in the infusion, where I was when the hospital was a concern. We did that for many individuals. In terms of whether or not it would help with COVID, that is something that's under investigation. We have no idea. So certainly, we've obviously we're looking at that now. And believe it or not, we had we were actually looking at a primary prevention IVIG study that had nothing to do with COVID in a different respect, but certainly we're going to be capturing that data and looking at whether individuals, whether it would help or not help in terms of COVID. We really just don't know. When we thought about whether or not for other reasons to hold IVIG, of course, we know that COVID sometimes can cause vascular issues and clotting issues. So, you had to take that into consideration because sometimes IVIG can do that as well. So you have to think about risk benefit ratios in that scenario. But certainly it's not that now that in New York, we're fine. We have people who are back on their IVIG and continuing doing what they're doing, but it's a risk benefit ratio, and we're learning about whether or not IVIG would be helpful in terms of reducing incidence of infection.
Dr. Dadwal, I'm going to get you another question, maybe in the data-free zone here, but okay, I know that if I'm immunocompetent and I get COVID 10 days later, unless there's a problem, I'm okay. I can start, hugging my wife again. But if I'm a CLL patient, when do I know that I'm not shedding the virus anymore? When do I know I'm no longer contagious? And I understand that the test can remain positive even when you're not really have active disease, because it could be measuring dead virus as well as live. So what are you telling patients who do test positive for COVID who are immunocompromised? How do they know it's safe again?
Yeah, this is a tough one. I mean, CDC has certain number of days they assign, 20 days or 30 days. But again, when it comes down to our immunosuppressed patients, every patient is different. Again, I would say how severely immunosuppressed they are. We've certainly had some patients with lymphoma actually who shed up to 90 days with a live virus. So definitely that has been reported. So I think in that situation, so what we do, at least at our institution, is we test them till they become negative, and we actually have access to a test on which we can sort of correlate with viral load by looking at what we call a cycle time. So, and there's some correlation, if your cycle time is below a certain level, you can sort of assume that you may have a viable virus. There have been studies done from Memorial Sloan Kettering, which have shown that correlation. So I think it is very, very individualized approach here too. For the most part, the guidance from CDC says that, for hospital isolation, that's where we, say if you are 20 days out, you can take them out of isolation. And so we sort of deal it in a little different way. We require 2 negative tests in a hospitalized patient, but if you're in the community, you're doing fine. I would still at least wait 20 to 30 days if you can continue to shed the virus. And if you have the access to the test, like at my institution where patients come in, they get tested and they go, and we can tell them that, you're likely out of the woods now, you can go ahead and hug and do whatsoever.
Practical question, can patients get the flu shot or the pneumonia shot at the same time as the COVID shot? Is there any data on that or the timing on that? I don't know if anybody wants to take that.
CDC just came out. I think ACIP came out and said you can give it at the same time.
Theoretically, I didn't think that you certainly given that we give lots of vaccines, some of them together for other vaccines, there's probably no problem. If you're concerned about if you're having a reaction to one or both and you want to separate them, I think that's fine. So some people are very nervous about what the reaction they might have to the COVID vaccine. So certainly if you want to separate your flu shot, thankfully, since we've been masking and washing our hands, we saw very little flu last year, but we're still recommending that people get their flu vaccines. It's just that obviously we did not see a lot of flu last year. But certainly if you want to separate your shots because you're worried about potential side effects and not knowing what the contribution is, I think that's completely fine. But there's no doubt, I think they're probably okay to get together as well.
Yeah.
We haven't talked much about monoclonal antibodies, and we're actually doing a full session on monoclonal antibodies. So I'm going to ask 2 questions here, and then I think we're going to have to stop to kind of give everybody a chance to kind of sum up and give final messages. The first question is, there seems now to be some shortages. Before there was this surplus and not enough people, and we at the CLL Society, all kinds were saying, know about these, get these. What's going on with that? How do you feel? And you don't have to answer this, but like I see states like Tennessee are prioritizing the unvaccinated, to get this. And I know that a lot of people, because of religious beliefs and other reasons, are not getting vaccinated, but have no problem getting the monoclonal antibodies. I just wonder, how do you see this? This is a limited, much more expensive resource. How do you see this being managed going forward? And where do you see these pre-exposed? I know we recommend that everybody, even if you're asymptomatic, if you test positive, you should get it. I just sent a friend who's got CLL, who's immunocompromised, and his wife is extremely immunocompromised for her lupus. Both of them were exposed because their son tested positive and lived in the house with them. They got the post-exposure prophylaxis, but it was a lot of paperwork, a lot of hassle to do it. Where do you see this going? Dr. Lamanna, you talked about the antibodies the most. You want to sort of handle that one?
I did, and obviously this is very difficult. And obviously this is gonna be dependent a little bit on regional variations, unfortunately, and very controversial. I think we clearly, I actually am recommending it for all my patients, regardless. Even though, as eloquently put before, is it necessary for all individuals? And obviously that spectrum of where a CLL patient may be on their journey and how their immune system may be different from one individual to another, but I don't think we have enough data. And so clearly, I'm concerned about any of my CLL people, regardless of not knowing what their real immune status, even if they're not on therapy. And so, at our institution, it hasn't been difficult because they call and literally we set them up very easily. They just come in and get their appointment for their infusion. But we understand that might be difficult where you are regionally and how that's going to be partitioned out. I do obviously, I think that those who aren't getting vaccinated, I think there should be we shouldn't be misusing that either. It shouldn't be just for folks who are obviously unvaccinated. I think people need to be vaccinated because that's the way, as Dr. Engel pointed out, the way to decrease variants and the infection is by having folks vaccinated. And so it's important that's available and people should get vaccinated. But we understand that obviously, depending upon limited supply, they may be, depending upon where you are, and how they handle that going forward is, I think, I don't know what else to say. It's tricky.
Yeah.
On a related point, not quite that question, but earlier on, Dr. Dadwal mentioned that immunocompromised individuals who have COVID shed virus for longer periods of time, and that even despite that, the recommendation is for mild disease but not hospitalized patients to get the monoclonal antibody. And as he mentioned, there is ongoing research. So it may turn out that even hospitalized patients with severe disease who are making a lot of virus would benefit from the monoclonal antibody, because what's happening in the immune part of the disease is not as well understood for immunocompromised individuals at the severe stage. So there may be a role for the monoclonal antibodies, even in severely ill hospitalized patients.
And to that point, Dr. Engel, we have used it in some of our CLL patients or lymphoma patients under emergency IND because it's not under the EUA. So to your point, we just don't know. There's not been much done in that field yet. But we go from case to case basis. They did not respond to remdesivir, Decadron. We tried all those things, and we reach out to the sponsor and to the FDA, and each case is evaluated, and those things, they have entertained that. So it may change in future.
That's a
And I
plug
saw
for
one,
clinical trials there.
Excuse me?
That's a plug for clinical trials for
Yeah.
sure.
yeah,
Right, Brian?
right. And I've seen one small study which showed that in patients who did not form antibodies, which would be us, and were critically ill, that the monoclonal antibodies did help those critically ill patients. I got to come up to closing questions. I'm going to keep this really short. If anybody if the person who give me the shortest answer, what are your thoughts on pre-exposure prophylaxis? It isn't there. We saw the AZD 7442 trial read out positively, more than a 3/4 reduction in symptomatic COVID, no deaths, no severe COVID. Is that the future for CLL patients? Let me give that to you, Dr. Dadwal. Do you have a thought on the pre-exposure prophylaxis for your immunocompromised community?
Makes sense.
All right, I can't ask for a shorter answer. All right, we're going to do a quick kind of, everybody has
Good
less
answer.
than that's a good, I agree with you. I think it makes sense. Dr. Dan Engels, any final thoughts? If you could keep it to about 30 seconds or so we can finish close to on time here. Yeah.
Sure. Thank you for having me. It's been a pleasure. I would say we should keep our eye on antivirals, which people are working on. There's one in clinical trials. I'm not saying that that's going to be the magic bullet. We don't know, but there will be, I'm optimistic, let me put it that way, that there will be over time antivirals coming out that can particularly serve immunocompromised population.
Dr. Stephens?
Yeah, I just want to reinforce that I don't want people to give up hope with this whole pandemic, because you can see, we have a lot more to talk about than we did a year ago. We have a lot more information about vaccines, about boosters, about more antibodies, more antiviral treatments. And so, although it seems, it still seems dire with all the infections you're hearing about on the news, I think there have been really significant improvements. And so, just continue being vigilant. And I think we are making some progress here.
Thank you. Dr. Lamanna?
Agreed. Testing's available. Vaccines are available, monoclonal antibody treatment hopefully available in most of your areas, but there much more that we did than last year. Patients are doing better. So yes, more hope for sure.
Dr. Dadwal, you get the last word.
I think I wouldn't repeat anything everyone has said. Well, what I would say is now behooves upon us to be more so at a community level. So all of us have a responsibility to do community outreach and urge the unvaccinated to get vaccinated. Because if you reduce the pool of humans who are not infected or who are going to get infected, that's where your variants are coming from, as Dr. Engel said. If you have a large susceptible population who is still going to get infected, we are still going to be in trouble. So I think it's about time as a whole, every attendant on this meeting, every participant in us, if each of us can influence few people, I think it'll go quite far.
Well, I want to thank all our panelists. This was an amazing discussion, and I certainly learned a lot, and I'm incredibly grateful to our panelists. And I think I hope I put them on the spot a little bit and made them a little uneasy with some of the questions, and I think they stood up to that, and I didn't just try to throw them softballs here, and I think that they really responded, I think, beautifully and nuanced to this stuff, and didn't just say, well, we don't have data on that, but did try to answer those questions. This wouldn't be available to happen without the support of from Adaptive and Janssen and Pharmacyclics and Bristol Myers Squibb. So we're very grateful for their supporting this virtual community meeting. Please fill out our post-event survey. That's critical for us to know how to do this. And we're going to be continuing to do COVID. We're doing a webinar that we'll be announcing soon that will be only on monoclonal antibodies. both for post-exposure, testing positive, and pre-exposure. That's a rapidly evolving situation, so stay tuned. On October 15th, we have a great webinar coming up, doing the right test at the right time. And this has to do, and Dr. Lamanna and I were talking about this testing, and it's sometimes what tests you don't get. Like you don't need to have a CT or PET scan at the time of diagnosis. So we're going to talk about all those issues and when you should get prognostic and predictive testing, and has that changed with the new targeted therapies. So we have a really great panel discussing all of that. And again, please let us know your feedback. Remind you that the CLL Society is invested in your long life. Please invest in the long life of the CLL Society by supporting our work, and there's a link there. The slides will be available to you with the video in the next couple weeks. If your questions didn't get answered, send them into our expert Ask the Expert program, and we'll try to get that back to you. But literally, we had a couple hundred questions, so there was no way we could get to everyone. So thank you for your understanding. Thank you for everybody's attendance, and thanks to our wonderful panelists. It was good to see some new faces and some familiar faces. Thanks so much.